So I'm going to tell you about the history of studying viruses, and this is something that I find really, really, really fascinating. The first virus was discovered in tobacco. Why at the time tobacco, this is late 1800s, tobacco was an incredibly influential cash crop. And because you couldn't sell diseased tobacco, it was actually very lucrative for the scientists to figure out a way to keep the tobacco plants from getting sick. So Dmitry Ivanovsky in 1898 looks at these sick tobacco plants and he filters their sap. And he's got a filter so small that it holds back the bacteria. And he finds even though the sap had all the bacteria removed, it's still infectious, which means something smaller than a bacterium is actually able to um, pass from plant to plant. The next big breakthrough happens in bacteria themselves. Hankin is in India at the time, and there is a raging cholera outbreak. And he finds that these patients, which are infected with cholera, which is a bacterium, when they drink water from the Ganges or Yamuna River, they get better. And so he starts trying to like filter this because he's like, what is in this water that is helping these people? But it's passing through the filters, even the fine ones. So he's not able to isolate that compound. Then you've got a man named Twart, and this is 10 years later. And Twart finds that his bacterial growth medium goes from clear to cloudy after bacteria are added. But when he adds specific bacteria into the strain, they result in the medium getting clear. Something is killing all these bacteria, and he calls it a bacteriophage. That terminology, phage means eating. So he said something's eating my bacteria. So it's a bacteriophage. So bacteriophages are viruses that infect bacteria. Basically, the way they're going to do it is the virus is going to bind with surface molecules on that bacteria. It's going to inject its DNA in there, and then it's going to use the machinery that the bacteria has in its nucleus that replicates its DNA to replicate the viral DNA. So basically, it's hijacking the machine and making the machine do what it wants, and then it's going to explode out and send all of its new viruses off to infect more cells. But this in and of itself is an interesting part in this history because now that we can study viruses in bacteria, bacteria are cheap. They're easy to keep around. They're easy to plate. So this means that it's suddenly really easy to study viruses. And the study of viruses explodes during this period of time. But the study of phages starts to halt, and I'm going to tell you why. You've got interest in it. 1919, Decherel, who is a French doctor, and his colleagues ingest the phages as a safety check. Basically, they want to make sure that it's okay to have phages. So they decide the best thing to do is drink them. Then they've got this patient who's a small child who has dysentery, a bacterial infection. The kid is dying, and they decide to give him the phages, and the kid lives. The research continues. Decherel went on and published this research about 10 years later. However, why don't we use phages today? Well, 1928, so this is before Decherel publishes his story, and not that long after us understanding phages, you've got Alexander Fleming coming along, and Alexander Fleming is a pig. And by that, I mean that he is messy in his lab. Fleming is like cramming his mouth full of lunch one day and puts whatever's left next to a bacterial plate that was uncovered and leaves on vacation. When he comes back, there's mold growing on the plate, and the mold has killed his bacteria. So Fleming discovers this mold juice, and he gets help from a buddy of his, and they purify this mold juice, and they find that it kills a lot of different bacteria. And now you've got antibiotics. By 1944, antibiotics are mass produced. And meanwhile, phages were still of interest to people, but they weren't properly stored for one of the larger studies. And as a result, this means that they weren't working. And since they weren't working, people are like, hey, we've got these antibiotics that work, and we've got these phages that don't work. We're going to put all of our eggs in that antibiotic basket. So does this mean phages are useless? No, they're not. They are more specific than antibiotics. The phage is actually specific to the type of bacteria it lands on. What this means is that if you're sick with staph, then the phage can land on the staph in specific, the infectious one, and it can kill off that infection. Meanwhile, leaving your beneficial bacteria out of it. Antibiotics, well, if you've ever been on a broad spectrum antibiotic, you know that it starts to affect your physiology because it is affecting your beneficial bacteria, specifically the ones in your gut, which is why you tend to get the runs. So this specificity is good because it means we're not affecting anyone that we don't want to. But the downside of it is that you have to know the exact strain of bacteria. So you can't just start someone on a course of phage. You have to actually do the hoof work. Another problem with phages is that they are viruses, and viral DNA is really quick at mutating. So theoretically, they can mutate into forms that are less good. And this means that they could pose a danger to people if we didn't understand how they mutated. So research on phages gets halted, and it gets all but halted until about 1980, and then it kind of starts to resurface. And in the 2000s, they start being used for treatment, but very sparingly. So this is becoming more and more important because during this period of time, that like 1980 to now, we started noticing antibiotic resistance, and that antibiotic resistance is problematic. Phages don't have that. So if you had an antibiotic resistant bacterium, you could still kill it with a phage. Now, meanwhile, in Russia, so US has sort of stopped phage research in about the 40s, really gone into antibiotics. And in the Soviet Union, you have communism, and they do not want any influence of the West. So you've got the Iron Curtain. Now, this Iron Curtain blocked a lot of influences, and some of those influences were from modern medicine. So they could not get the antibiotics that we had in the West. Instead, they had to do something else. What do you think they did? Ha ha. They invest in phage therapy, and phage therapy is widely used in Eastern Europe, even to this day. So interestingly, because you have that the phages being used in Europe, but the Eastern European journals are writing in Russian, and the Western European journals are writing first in German and then predominantly in English, you have the scientists not even talking to each other. So the Iron Curtain is preventing medicines, but also preventing the science from going both ways. And this divide still is impacting the science, which is fascinating. For example, if I asked you if you would take a virus that's going to make you better from Russia, I don't think you would right now. And it's because we have this sort of politically contentious relationship with them. So people fear the phage, right? They don't want to get injected with a virus. They don't like that idea. But we've got to think about how to overcome fear. And the best way to overcome fear is through information. The more informed you are, and the more that you understand how these things work and how specific they are, the less scary it could be. And to me, the idea of something that's not going to kill off my gut bacteria is much more intriguing. Now, let's take a minute and let's look at how viruses have been used for good, because we've been thinking about viruses for bad for a long time, especially lately. But viruses, what they do is they deliver their genetic material to your cells, and they use your cells to make more of their genetic material. Well, they can be gene delivery systems, and they've been used in gene therapy to deliver genes into human cells to make up for faulty genes that are in those cells. This has been studied since 1950, and gene therapy has been going on since 2000. And more recently, viruses have been used for cancer therapy. And some of this use has been to get your body to recognize the cancer cells as invasive so that your immune system will fight them off. So they're not all bad. So at the end of the day, when you think about it, these phages are kind of the up and coming therapy. They're nothing to be afraid of unless, of course, you're a bacterium.